All right, very good morning, everyone. So we are currently doing the student project presentations, and as we have already learned about different type of proteomics data analysis, idea was how you can take some independent data set and take that forward and do analysis in your own way. Just to remind you that presentations are worth 20 marks, which is like doing a project and presenting in the class. Each day class activity along with the write-up is going to give you 2 marks, and therefore all the subsequent presentations are going to give you total 18 marks. Presentation is 15 minutes and 3 minutes will be for Q&A. And all the students are encouraged to write the questions in the chat box, which presenters should reply, and they can address those questions. After the lecture is done, once I upload the video, in the description of the video, you will have Google form where you need to do the summary write-up as well as some MCQ questions which will be given to test your understanding of those presentations. So I'm sure we are all enjoying these presentations and you are taking notes and understanding the lectures very well. Just to give you some more points of clarification, we have informed the schedule of these presentations, and that presentations are going to happen quite ahead of time in the beginning of the class as well as just before midsem. Post midsem, 2 lectures were just scheduled to give you some hands-on activities and do the projects for you. Further, I have also given in-person meeting slot to people who are ready for this week presentation, so that if you face any problem, you can talk to us and I can help you, or our team and TS can help you to do your projects easily. That's what we have scheduled a meeting early this week. And many of you have joined actually for that meeting as well. So if in case anybody is sick or having any serious medical problem for which you have a genuine medical certificate, and if you are able to produce that, then only please make a request for rescheduling presentation. Otherwise, it is very much logical and possible that sometime you are stressed, sometime you are short of time, but those are not the genuine reason to ask a last minute request to reschedule the presentation. Because every class we have assigned certain student task and the whole class time is already being planned in such a way. If you inform me yesterday night or today morning that you cannot make presentation because of some other reason, it is not possible for us to find a slot for somebody else to assign that. Additionally, it is also if I take a new topic to teach you, how I can fill that topic in 20 minutes, 15 minutes kind of time, and without any flow of the continuation of that topic. So therefore, that also is not possible to fill that time wisely with our own lectures per se, right? So you have to understand that you have to be professional. You have to plan the things in such a way that you have to make these presentations on time. Please do not request for any compensatory grades or marks for these presentation, which we will not do that. If you're missing out your slot, you're missing out your slot, and 20 marks for that matter, because we have been assigning these things ahead of time. It is a large class activity where if you are changing the slot, then you have to be very serious about it, why you are changing the slot. In case you feel you have a friend who can swap your slot, that is possible. You can swap a slot with your friend, but you can inform us ahead of time so that we can reassign the slot. But if we have assigned 5 students for today's presentation, we are expecting all the 5 students should make the presentation unless somebody has a valid medical reason for which they are giving a certificate, and we have to vet that with our CMO from IT Bombay. In case you are facing any problem in recording video, that may be logical. You can also make a live PowerPoint presentation, which is possible in the given time of 15 minutes. I hope we are very clear about all the guidelines, which we have been reiterating again and again. If anybody has any problem, any questions, anything unclear, please ask me right now. I think this is the time to discuss this point before I start today's presentation. Anybody is unclear or something is not conveyed to you earlier and something is not clear to you, please let me know so that we can clarify your points. I hope you understand this is a activity. If 2 people don't present on a given day, it is going to derail the whole class time, and we are going to get short of time at the end. A lot of other lectures have been planned: microarrays, surface plasmon resonance, other biosensors, multi-omics. Many things are yet to be covered, which I have planned for finishing your presentation after that. If we delay the presentation because some people are not able to present, then it will be difficult for us to reassign those. So we have to take just call, either you present or don't present in the given slot. If you are able to As I said, even if you are not able to completely be prepared, which many times you are stressed and for some reasons, still you should make an effort to give your best and don't get the things delayed, because otherwise you are dragging all your different class activities, assignments, and even overall semester activities in that matter, and you'll be more stressed in that way. We are there. I have been keep telling you, I have assigned at least 2 slots for meeting with the student this week who are due for the presentations on Wednesday and Friday. Just so that if you need some help from our team, and many of you ask help, and our team has given you help of doing your data normalization or giving you the right strategies, we are there to help you out. We are there to make your presentation to the best possible quality which you need from our side. But please don't ask for any excuse and later on the scaling up of the grade and compensatory grade for this activity, which is not possible. I hope you understand. This is the rule for every student, and whole class have been informed ahead of time since the beginning and have been reiterated again and again. So with this, let me now start today's presentations. I hope we are all clear about it. I have also put this in the things in the email, and I'm just repeating myself in the class here. So the first recorded presentation which I have is from Nilesh. Although we have multiple presenters here, but I'm first starting with Nilesh, who is going to talk about global proteomic analysis of glioblastoma and looking at the effect of some mutation on IDH. So let me start his video, and Nilesh, please be ready to do the Q&A.
Good afternoon, everyone. My name is Nilesh. I'm first year PhD student at Chemical Engineering Department. Today, my topic of presentation is global proteomic analysis of IDH positive versus IDH negative glioblastoma samples to decipher the biological alteration. In the most common primary brain tumors are glioma. Among all gliomas, glioblastoma is most common and most aggressive tumor. The median survival is 14 months, which means after the initial diagnosis, within 14 months, 50% patients died. In gliomas, the most common mutated protein is IDH1. Now, we can see in normal cell, this IDH1 has a role in TCA cycle, where it converts isocitrate to alpha-ketoglutarate using NADP plus and also forms NADPH. But in tumor, it not only stops at alpha-ketoglutarate, but it also converts this alpha-ketoglutarate, like alpha-KG, into 2-HG, the new oncometabolite. Now this, while it's converting alpha-ketoglutarate into 2-hydroxyglutarate, it consumes NADPH and forms NADP+. The total pool of NADPH gets reduced in IDH mutant tumor. Now this NADPH has role in lipid metabolism, which we'll see in later. The other tumor characteristics like proliferation rate, invasion, and necrosis of IDH positive tumor are lower compared to IDH negative tumor. Now here, what I mean IDH positive and negative. IDH positive means tumor having mutations in IDH, while negative don't have any IDH mutations. Here we can also see the IDH mutant tumor having low lethality compared to high. From the survival graph, we can see that the median survival of IDH mutant tumor is 300 days, while in IDH wild type patients, the survival is approximately 800 days. Now, my workflow. So I get data from tissue samples of glioblastoma patients. Now, from these tissue samples, the proteins are extracted using urea buffer extraction method. And these tissue samples are analyzed for IDH mutations using sequencing. The raw data and the proteomics raw data was analyzed in MaxCon, and I get the processed file. I use this processed file in MetaboAnalyst to perform correlation plot, missing value imputation, statistical analysis, differential expressed proteins, and pathway analysis. For missing value, So for my data, I found 14% missing values were detected. From these 14% values, I performed missing value imputation. So I removed 30% features with having 30% missing values, and missing values are imputed using KNN feature-wise method. After that, I performed log transformation of my data and median normalizations to reduce run-to-run variation. Here we can see my data before log transformation after median normalization. Then I performed principal component analysis to see if there is any outlier present in my sample, like bad sample or anything. Here we can see all samples are present in their respective compartment. So I didn't exclude any samples from my analysis. Then I performed VIP score and correlations to get an idea about outlier samples. Here, for correlation, I kept 0.5 as correlation value, and I didn't see any outlier. So again, I moved again to the fold change and t-test. For fold change, I kept my parameters as 2.0 as significant fold change, and for t-test, I kept group variance as unequal, raw p-value at 0.05, and the samples are present in the order of IDH mutant versus IDH wild type. Then I plot the volcano plot. In volcano plot, we can see that there are 24 genes are upregulated in IDH mutant glioma, while 28 genes are downregulated. Here is the just graphical representation of BB607-Proteomics-20211001-Lecture-17 protein, which is upregulated in IDH mutant glioma. Then to see the visual representation of my proteins in differential expression of my proteins in IDH negative and IDH positive tumor samples, I plot heat map. Here we can see these proteins are upregulated in IDH mutant tumor while downregulated in IDH negative tumor, and vice versa for this protein. Then I perform the gene list I obtained from volcano plot is used for reactome pathway analysis. In reactome pathway analysis, I found that the many pathways are dysregulated in IDH mutant glioma, but major pathways that are dysregulated are mitochondrial fatty acid beta oxidation, glycogen-related metabolic pathway, and glutamate-glutamine metabolism. From my normalized file, after metaboanalysis, I used that file to perform gene camera-based reactome pathway analysis. Here we get some pathways which are significantly upregulated, while some pathways which are significantly downregulated based on gene set expression analysis. So the top downregulated pathways are here also related with fatty acid metabolism, mitochondrial fatty acid beta oxidation, glycogen storage synthesis, and metabolism related pathways. While the upregulated pathways in IDH1 mutant tumors are cell cycle control pathway and the translocation of GLUT4 to the plasma membrane. Now this GLUT4 is a glucose transporter. We will see what its roles has in cell. After literature review, I found that under normal circumstances, IDH activity reduces NADP+, while catalyze the oxidative decarboxylation of isocitrate to alpha-ketoglutarate. Here we can see that in mitochondria, isocitrate is get converted into alpha-ketoglutarate. Also in cytoplasm, isocitrate is get converted into alpha-ketoglutarate via consuming NADP+ and converting it NADPH by enzyme IDH1. But in IDH1 mutant tumor, this alpha-ketoglutarate is gets converted to 2-hydroxyglutarate by consuming NADPH, so reducing NADPH pool of total pool inside the cell. The glutamine is used to now in IDH mutant cells, the alpha-ketoglutarate, which is a key metabolite in a TCS cycle, it's get depleted. So to replenish this alpha-ketoglutarate, Another pathway gets activated where from glutamine, the glutamate is formed, and from the glutamate, alpha-ketoglutarate is gets synthesized to remain like constant alpha-ketoglutarate flux inside the TCA cycle. But the lower concentration of alpha-ketoglutarate and NADPH impairs the fatty acid metabolism. So here we can see that because of the NADPH gets consumed in the gets consumed here. So the NADPH total NADPH pool inside the cell is get lower. So for fatty acid oxidation, beta oxidation of fatty acid requires NADPH, and now due to the reduced NADPH, there will be no fatty acid oxidation. So cells cannot use fatty acids for their energy requirement. Now the only source cells have to perform the fatty acid to get energy is glucose. So that's why in IDH1 mutant tumor, the glucose GLUT4 transport pathway is increased to get increased glucose inside the cell, and from that glucose the acetyl-CoA is get formed, and now this acetyl-CoA is get converted into citrate. Now then this citrate is get converted into fatty acid synthesis to form a fatty acid, and now this fatty acid is used for cell proliferation. Now again, after that I performed to compare my data with TCGA data. I take a cBioportal IDH mutant and wild type patients data and perform differential expression gene analysis. Whatever gene list I got from this TCGA data, I compared it with my metabolic analysis gene list. I didn't see any gene matches with reverse phase protein assay. The data I got from reverse phase protein assay. In reverse phase protein assay, we know there are very less proteins which are used to analyze the tumor sample. That's why we didn't see any hit in reverse phase protein assay. But from transcriptome data, I identified that 34 genes from Metabo analyst gene list are present in TCGA data also, which means this data these two genes are these two data sets are complementary to each other. While the 18 non-matched genes which are present in metaboanalysis gene list, the 3 of them are mitochondrial genes. And because of these are mitochondrial genes, these genes are not present in TCGA data. So here we can say that using our proteomic analysis, we find novel 3 genes, ECI1, ECH1, and PDP1, which is differentially expressed in IDH mutant and IDH wild type tumor. In conclusion, in IDH mutant glioblastoma, due to increased oncometabolite 2-hydroxyglutarate, the beta oxidation of fatty acid is impaired by low NADPH concentration. While using proteomics, we identified unique mitochondrial genes ECI1, ECH1, PDP1 as differentially expressed genes in IDH mutant compared to IDH wild type glioblastoma. These genes we can only be identified using proteomics assay because in gene expression analysis, these genes gets lost during sample preparations because these genes are transcribed by these genes are mitochondrial genes. These are references. Thank you.
All right, Nilesh, you are there. Can we talk to you?
Yes, sir.
Okay, so yeah, I think it is an interesting presentation, and the topic itself, just for everybody understanding, is not very old. Of course, the glycolysis and TCA cycle, but the role of this important gene IDH and its mutation was only revealed in 2008 when there was a large genome sequencing project was done by TCGA, the Cancer Genome Atlas, right, which Nilesh should have read. And that actually showed the power of these kind of omics technologies, that how these cancer like glioma and glioblastomas, which are very deadly tumor and not being understood well from the decades, just simple sequencing technology revealed IDH1 and the mutation can be so important. Our lab also works on the glioblastoma and gliomas, and we work very closely with many doctors from Tata Memorial who work on the patient's surgery for the glioma tumors. And what we see, the change because of IDH has become rapidly. Like, the IDH has really come to the clinics, including the Tata Memorial, where all kind of classification of grading and everything has changed now based on the IDH. So think about one sequencing project, one gene role can be so important, and even the entire WHO classification scheme of the glioma tumors has been reworked on based on the understanding of which ones are wild type, which ones are mutant of IDH1 and 2. And now these are being routinely sequenced for all the patients, irrespective of their other features. So this is very important. And as you have rightly then worked out on the workflow, now I can set up the timer and talk to you about some questions with respect to your presentation. Are essentially that the missing value was, I think, less in this case, right?
Yes.
So less than 20% probably, right? So one could always consider even that, whether you want the data with the missing value imputation or you do not want. For example, if the missing value is less, you might want to focus only on the data set which is having all the values, right? So one could also look at how much data will you lose if you are not doing missing value imputation. So that could have been also considered as the way forward for the analysis. Nevertheless, you use the right way of doing KNN and doing the imputation for that. Your correlation plot also revealed that the samples are quite homogeneous and there was no problem. You showed your PCA versus Plsda plots. What the difference is between these two, like when you showed those two Venn diagrams?
Sir, there is a difference in sample correlation in two different ways. In PCI is a simple principal component analysis, and in PLSDA they use like slightly different algorithm which clearly identifies like differently correlated genes samples.
All right, so just read a little bit statistical reasons behind these principal components and partially discriminant analysis. Okay. Then I think you have talked about the major finding from your analysis was essentially in context of glucose transporters and the fatty acid oxidation, right? How
Yes.
they are so crucial for the IDH mutations. So if you were to take this experiment forward based on your understanding and literature review, what kind of additional experiments can you perform to validate your hypothesis?
Sir, here I see like I compared my hit genes with TCGA data. So TCGA data is a gene expression data, and we have protein expression data. So when I was analyzing these two data sets with one data set with another, I see like many of the genes are gene expression data is correlated with protein expression data, but there are total 9 genes which are showing opposite correlation with their gene expression. What I mean is like they are highly expressed, like their gene is highly expressed in IDH1 tumor, but their protein is down-regulated in IDH1 tumor.
That is okay. Let's say you are looking at multi-omics analysis and looking at gene versus protein correlation. I'm saying even if there is no correlation or you are finding something, the trends are different. Now you are still finding an important role for these pathways, right? And you want to understand it better. What else you should do now? Whatever you have done, you have already done. What else you can do to validate understanding of these particular pathways?
These pathways we can use, sir, C75 inhibitor, which is like lipid synthesis inhibitor. Here we say that already we know for cancer, lipid synthesis is major. Lipid synthesis has major role in cancer cancer proliferation rate. So we can downregulate the IDH1 mutant proliferation rate by using C75 as an inhibitor.
So good idea. And likewise, so I think you can choose different inhibitors linked to these pathways and look at in the different cell line of these patient-derived cells or the other available cell lines and try to understand the role of them. Also, one could think about even doing some enzyme assays for measuring these, fatty acid oxidation levels and looking at different glucose-related metabolism. You can measure those easily, right?
We can also measure, sir, 2Hg levels in tumor samples.
Absolutely. So those are the things which also we should think about biochemical ways of validating our findings. Let's not only rely on the very specific gene and protein correlation. Also, one last thing is, your time is already up, that RPPA, what does it stand for?
So RPPA is reverse phase protein assay, which is like
arrays.
Protein array.
Correct. So that's where you mentioned assay in the presentation also. It is actually arrays, and you look for
where we embedded antigens of particular protein, and then we calculate the total number of different proteins which are differentially expressed based on their fluorescence index.
RPPA, you actually take, let's say, patients' tumor biopsies, right? So if patients are having the tumor chunk of the tissue, you take the small tissue biopsies, print on a chip, and now you are going to have antibody for IDH1 and screen that where you see the signal. So this is why it is known as reverse phase protein array. I will talk about that in the microarray section when I come to that. But yeah, these are the way the findings can also be easily taken to the clinical settings when clinicians are doing these kind of tests right now. Yeah. All right. One last question. One more thing which came to mind was essentially one protein which you showed in your upregulation very nicely after metabolite analysis.
Yes.
BIP plots, which was P42126. What is the name of that protein?
Sir, I have another file. I can write it in chat.
So make sure in the presentations, we should not give the accession IDs or protein IDs, but rather also give the protein name, because so that we can relate to what that protein means, right? If it is interesting. And also we should know about the role of that. Yeah. All right, Nilesh, good start. I think that's all for you. Okay.
Thank you, sir.
Sounds good. So let me now go to the schedule for other presentations and other presenters. All right. So is Balivada Sahasriya, are you there? Next is a student who is lined up. I have not received any response, not the video. Next is Balla Sarat Chandra. Are you there?
Sir, I'm Sahasriya. I actually couldn't complete my presentation, which is why I didn't do it.
Well, I have already clarified myself, what should have been done. Right? And please understand whole class timing is being used right now. So last minute, and if any one of you need any help from our side, we are always there to help you out, to complete your projects. But last minute change will be problematic. So we have to drop you right now on this. And Sharat Chandra, are you there? Balla Sharat Chandra. Bodapti Yagneshwar, I think I received some email from you, but are you there? All right. Chinati Narzare, are you there? Okay, so I hope you all understand that it is very difficult if several people drops in the last minute, and we have no way to actually become, very much considerate about this problem right now. As you understand that schedule has been made for the whole class, and people have to be more professional to do these things. If any one of you are available here and you want to make a presentation, please do that. Even if you're not able to upload a video ahead of time, that is, we can waive that off. You can make the presentation right now. There is no possibility for compensatory presentation because the next class is again next 5 students are lined up. How can we make 7 people presenting in the class time? Any one of you want to make presentation? Please let me know right now. All right. If anybody want to say that, you can raise hand or write in the chat. What I have planned now, in the meantime, we are now going to continue with a presentation which is going to be by one of the pioneers of the proteomics field. His name is Dr. Bernard Kuster. So Dr. Bernard Kuster, he is one of the pioneer scientists of proteomics who has developed the first draft of the human proteome map. This happened in 2014, first time when the using mass spectrometers, people have showed that how you can use the mass spec to confirm that all the peptides and proteins for the human are actually can be experimentally tested out. Experimentally, there is evidence for that. And that was a really big finding because so far from the Human Genome Project, we have idea for theoretical possibilities of the possible proteins, but we have no idea that that these proteins actually exist or not. So therefore, we the first time 2014 there were two groups, one from Germany, Dr. Bernard Kuster and his team, Other was Akhilesh Pandey, his team from Johns Hopkins and IOB. They simultaneously did this work and published in Nature, showing the draft human proteome map. After that, Dr. Bernard Kuster and his team has been very creative in generating a lot of good software, including Prosit, which is very actively used for the targeted proteomics and verifying your sequences. And also then his team is working on various type of synthetic peptide and novel ways of doing mass spectrometry-based proteomic analysis, and especially also some of the native protein characterization, trying out different things with the liquid chromatography, how that can be useful for increasing your coverage. So we had invited him for a talk, which I'm going to play right now. And in today's write-up section, you will have some MCQ questions based on Dr. Bernard Kuster's presentation and of course Nilesh's presentation, both of that. So I'm going to start this presentation. Please watch it carefully. At the end of the class, we will have a Google link, Google form a link to you, where there will be several questions based on the Dr. Kuster's lecture will be also asked.
Ladies and gentlemen, it is our great privilege and honor to have amongst us one of the pioneers in the field of proteomics research, Professor Bernhard Kuster. He joins us from Technical University of Munich, Germany. Professor Bernhard Kuster obtained a PhD in biochemistry from the University of Oxford. Thereafter, he completed his postdoctoral training, first with Professor Matthias Mann at EMBL, Heidelberg, Germany, and at the University of Southern Denmark. Professor Kuster is the chair of the Technical University of Munich Department of Biosciences and a member of the Graduate School Experimental Medicine. His research is funded by the Center for Integrated Protein Science, Munich, the German Consortium for Translational Cancer Research, and also the pharmaceutical industry. Between the years 2000 and 2007, Dr. Kuster worked for the biotechnology firm Cellzone, where he held several positions, including that of Vice President of Analytical Sciences and Informatics. In the year 2007, he was appointed full professor and chair of proteomics and bioanalytics at Technical University of Munich, Germany. Presently, he is the head of the Department for Biosciences and a co-founder and co-director of the Bavarian Center for Biomolecular Mass Spectrometry. Dr. Kuster is a member of editorial and scientific advisory boards of several journals, university institutes, and companies. His current research interests include functional proteomics, chemical biology, cancer, biomarker, and drug discovery. Please join me in welcoming Professor Bernhard Kuster.
To present a few stories from my team who's been doing all the work, and it's fun to have all these great young people around me that basically as a team accomplished what we do. But before I start, I should make a few quick disclosures. I founded 2 companies. I've got nothing to do with their daily business, but I'm a founder and shareholder. We do currently get some research funding from Merck, and IBM, Thermo Fisher, and SAP are supporting us in kind with some of our technologies that we build and run. So that has hopefully nothing to do with the content of the science other than it makes it better. All right, so just as a background from my lab, we basically do kind of 3 things. And one is to map out proteomes, because I think that is an important area just to know which genes actually exist as proteins, where they are in an organism and in what quantities and how they are modified, and so that we might better understand what they do. The second pillar of the lab is actually understanding how drugs work, with a particular view to precision in medicine and oncology. And the third pillar is building proteome tools. And this is really the content of my talk today. And I thought for a workshop on proteomics, this is more fitting than giving you a high-level story about human or plant proteomes, but actually get down to the dirty details of every day's lab business and what we could do there, because it's true to say, I think, still that proteomics is a technology-driven area, and we haven't seen the end of these areas yet. So what I'm going to talk about is one thing in quite some detail, which is proteome analysis by micro-LC-MSMS. That's an area that I find extremely exciting at the moment. And the second is to look at FFPE material analysis using nano LC devices coupled to FAMES and MSMS. And I'm just going to hint briefly on an informatics tool that we've built called ProZit, but you're going to have a full talk about ProZit or our IT tools on Friday when Matthias Wilhelm is going to talk about this. So straight in, one of, let's say, the most exciting technology developments in my lab, at least, has been driven particularly by Yang Bian and also assisted by Florian Bayer, where we basically took a look at solving all the problems with nanoscale liquid chromatography, because many of you will know that this comes with a lot of issues. And inspired by the work of other people, in fact, we've started playing around with the idea of using 1 millimeter micro LC columns for some simple reason that you wouldn't worry too much about how much you need to load, and the chromatographic performance should actually be great. And what a lot of people in our area forget is the mass spectrometer can be a great device, of course, but if we do poor chromatography at the front end of this mass spectrometer, it's not going to be able to compensate for all of the mistakes we've made already. But just to put some perspective as to how that sits in terms of scale, the typical nanoscale LCMS is done 75 micron ID columns, and what we're going to talk about is 1,000 micron ID column, and the relative sectional area is about almost 200 times bigger than that of a nano, and that comes with a couple of clear advantages. But just to illustrate why we even started this, you can see here two LCMS traces of human plasma digests, and the top one is the nanoscale and the bottom one is the microscale. And you can see very clearly that while we have a lot of problems here up here with the peak broadening, we can generate very, very sharp peaks using a microscale separation, and that gives peak capacities of around 700 within 45 minutes of LC time. So this simple characteristic led us to investigate this a little more, and essentially what we found for plasma is actually true for other body fluids as well. And what I should mention is, and maybe for the proteomics community that may come as a big surprise, we run the chromatography at 50 microliters a minute, which is a lot more than the typical 200, 300, 500 nanoliters a minute that you would do for a nano system. But you can see either for CSF or plasma or urine, basically all of these produce very sharp chromatographic peaks. And with CSF, we can get within half an hour gradient, 700 proteins, 250 for plasma, 1200 in urines, with loadings that are, for these body fluids really are not a lot. It's very hard to, even from a drop of blood, it's very easy to get a 20 microgram of plasma protein from a drop of blood. And what maybe is an even more important feature is the carryover from run to run almost doesn't exist anymore. So we have almost no carryover between runs. That is a great asset when doing proper chromatography. So and here's basically a picture from the paper we initially published on this idea. When you look at the chromatograms down here, the one in red is a microscale LCMS, and the blue is the nanoscale LCMS. And you might appreciate that in a 15-minute gradient, the micro LCMS is already over before the very first peak on the nano system has come out. So we can easily double the throughput here with very little downside in terms of sample loading or also sensitivity. But this is certainly one thing to keep in mind, because we run at a much higher flow. The microsystem is about 3 to 10 times less sensitive than nano LCMS. So it will not be applicable to each and every application, but it will be applicable for a lot of applications. And this is what we try to show here in this table. Whenever we do full proteomes of cell lines or tissues, where you have 100 or 200 micrograms of total protein available, it should not be a problem at all to get to very competitive numbers of proteins and peptides compared to even often 2D LCMS approaches. So with that in mind, we also wanted to see if we can sort of stress test that system. And Yang came up with this rather elaborate way of putting together 1,500 consecutive injections that consist of 10 cycles of 20 HeLa digests each, followed by some QC peptides, followed by 20 urine samples, 20 CSF samples, a couple of plasma samples, placenta samples, and then going around that circle 10 times. And essentially that was a total of 40 days of machine time. And we basically asked the question, how robust is this? And it turned out to be extremely robust. When you first look at the left-hand side of this plot, what you can see here is the elution times of synthetic peptide standards that we use as retention time calibrations. And when we run them alone, we get a certain, of course, retention time characteristic. And when we run them spiked into the body fluid, we, of course, hope that we will detect them at the same position. And you can see from this correlation plot where each of the dots here is a cloud of dots of many replicates of detecting these peptides, you can see that these two measurements correlate extremely well. So the slope of this is 1.0, so we're not getting any particular shift, and the intercept is 0.01 minutes, which is barely a second. So essentially that says the chromatography is so good that no matter whether we have a sample on the column or just a QC, it doesn't change the retention times of these peptides at all. And the zoom in is shown here that even though these two QC peptides, they only differ in a few seconds of elution time, they are nicely separated by this system. And that chromatographic performance directly translates also into quantitative accuracy. So when you look at the coefficient of variation of the different types of samples we had in this stress test, you can actually see in this cumulative density plot that the vast majority of all these proteins is quantified with a CB of less than 10%. And that is, I think, quite interesting because it makes these measurements fit for many applications in the clinics, and something that is not really new to people who analyze plasma samples, but here are repeat plasma analysis of 5 individuals, and the t-SNE analysis very easily pulls those individuals apart, saying essentially that the plasma proteomes of individual people are different to each other. So we can now robustly measure this, and one of the so far unpublished stories is that, well, this is should also be high throughput, and actually it is. So we've gone through almost 400 cancer cell lines, and we're in the process of scaling that to more than 1,000 at the moment. And for each of those, we get in excess of 10,000 proteins for each cell line, and this is how they distribute. And for good measure, we've also looked at the phosphor proteomes, and you'd see that it is more variable here, but that is actually measuring biology. And here on the cluster heat map here, the only point I'm trying to make is that you can't really see anything, and you have to take my word for it that the 20 QC runs all cluster next to each other, essentially saying that we can now really cluster data according to what we measure rather than the device that is used to measure it. And that used to be a big problem. Okay, so we have good reason to believe that this is high throughput, but is it also robust? And that is something which we've just typed up again, and Yang will have that paper out in Analytical Chemistry in a matter of days now. So what we've basically, since we started doing this in 2018, we've kept doing it. And so what you see in this chart is here is the number of analyses that were run on this microflow LCMS system, either on an HFX or on a Lumos, over the course of about 2.5 years. And by now, I think we are standing at more than 38,000 runs and still counting. So that's good for robustness figures. And just here, this pie chart gives you an idea of what types of samples these are. For the Lumos, we do a lot of high pH fractionation and read that out by LFQ quantification, but we also do a lot of TMT, and this is just for you to see how sort of these samples distribute. even though some people may think for proteomics it's crazy to inject 5 or 10 or even 20 microgram of a digest, for these columns this is actually very little loading. And that translates into a huge column robustness as well. And this is shown in the pie charts here. So on our QEH of X, one column has by now seen more than 14,000 injections on the same column, and on the Lumos it's 12,000, and both of these columns are still on the machine and they're still running samples. So that is obviously very good for any application where you want consistency in your analytical system. And if you have 5,000 samples to run in a project and you can do that on a single column, you're going to take out a lot of that variation that you usually have in the nano LC world, where a column can die any moment. And that is also illustrated here in this picture here. There's again these QC peptides that we ran sometime in 2019. You had nice separation of the two, and then something bad happened. There was a sample injected that partially blocked that column. So actually Yang turned it around, and we then started running it in the reverse order. And you can see that the retention time shifted a bit, but the separation was nice. And then more than a year later, almost a year later, he turned it around again to the forward direction, and the separation was still great. And now even the retention times returned to almost identical figures to what they were almost a year earlier. So injecting little into chromatographic columns is good for the lifetime of your column. It's good for separation. It's good for lifetime. It's good for data consistency. And this is just a different way of illustrating this here. Here is a couple of experiments using different gradient times from 10 minutes to 2 hours, run in 2018. And what is plotted on the y-axis is the full width at half maximum of the chromatographic peaks. And for 10-minute gradients, they are sort of the median peak width is about 3 seconds, and it goes up, of course, with longer gradients to about 10 seconds. And this is the same samples run 1.5 years later, and you can see that the distribution of chromatographic peak width is pretty much identical. And this is also the case when you look across single gradient, but many different, always the same sample, the QC samples that we run on HeLa digests across a large number of runs across an extended period of time. So that really convinced us that this is also robust. And again, returning to the example of plasma, if you have a nano separation, it's very common to see these very sort of pigtailings on the late side of the chromatographic peaks. You don't have that on the microcolumns, even though the loading here is 30 times different. And just to illustrate this again by zooming in, if you were, the blue line here is the micro separation, the red one is the nano separation, and it's not hard to appreciate that not only the blue peak is sharper, but also has less tailing. Okay, sorry for the bad quality of the graphics here. It's just a copy-paste problem, I guess, from a PDF. And Yang has also asked the question, well, can we actually use the system to go very deep into a body of fluid? And the answer is yes, you just need a little more time. So if you spend about 1 day of machine time, you can get to 2,500 plasma proteins or 5,000 proteins in urine. And he's just highlighted a couple of cytokines and chemokines that could be detected in this way, which are usually considered low abundance samples. So switching gears a bit, so we've convinced ourselves that that this system is both high throughput and also offers high robustness. And now the latest on this is still unpublished, but Yang also then asked the question, so what about doing proteomes at single shot? And a lot of people are trying to do this because, again, if you can simplify your workflow, including the sample preparation, the LCMS, the data analysis down the road, the hope is not only to be able to generate more throughput, but also better data consistency, because every step you take out from a workflow should lead to less variation. So what we've asked here is, well, how far can we push the idea of single-shot proteome analysis? And the data that you see here in this slide is just counting the number of peptides and proteins as a function of injecting more and more and more digest into such a column. And measuring this either with the 28 or 41 Hertz mass spectrometer settings. And you may say this is a crazy amount of protein digest, but for those of you who are used to running Western blots, I mean, these are typical loadings here for a single well of a Western blot. And then you probe with one antibody or sometimes 2 or 3 to detect a particular protein. And if we do this with mass spectrometry, we can detect 30,000 peptides or close to 5,000 proteins that way. So that gave us some encouraging results that single-shot proteome profiling might actually become possible. As a point of detail, you can see here that the slower mass spec method actually outperformed the faster. And the reason for that is simply that if you run the mass spectrometer too quickly, then you have too few ions that actually are being detected, and the quality of the tandem mass spectra goes down so that you can't ID them any longer. And we'll return to that in a moment. So what we then also look at, obviously, again, is the chromatography. And what you can see on the left-hand side, as you load more sample digest onto such a column, what you observe is that the peaks get shifted to earlier retention times. This is not at all a surprise. This is what just happens if you have a lot of stuff loaded at the top of the column. So you basically get some displacement, but the peak shapes in chromatography are still very good. Another thing that you see is that at some point the whole system saturates. And this is not the chromatography, but it's actually the mass spectrometer. And that is apparent from plotting the actual ion current going into the instrument. And in red, you can see the MS1 intensity really begins to saturate beyond loadings of 50 microgram. In MS-MS, you don't see that so much because obviously the ion current after fragmentation is always much lower than it is for the intact peptides. Okay, so we basically also what that says, loading more than 50 micrograms also really doesn't make much sense, and we're back to loading sort of Western blots with similar quantities. Now, where does that go when we now look at the gradient times? Obviously, one would expect to get more peptides and proteins the longer the gradients become. First of all, here on the left-hand side is a quick look at the chromatography. And these box plots basically, again, say how wide are the chromatographic peaks. Within 1 hour, we have about peaks that are 6 seconds sharp. And if we extend that to 6 hours, they become much wider. And in the inset is shown how the peak capacity develops as a function of gradient time. And we can see we can push it to nearly 1,000 peak capacity, but the increase from 1 hour to 6 hours in total is only about 50%. So one has to really think about how much time one wants to spend for a 50% increase in peak capacity. But what is interesting also is in the middle, when you, again, now look across the gradient times and ask the question, how many sort of precursors can we actually detect in MS1? We can see that it's steadily going up. And how many of them can we try to fragment? And this is a relative scale always to 1 hour gradients. You can see the mass spectrometer is really trying hard to fragment many, many of those peaks. So it kind of triples the number of peaks that it selects for MS-MS as the time gets longer. But the blue line here really is what is the result of this. There seems to be a sweet spot at around 3 hours where the largest number of peptides can actually be identified. So you might ask the question, so why does it go down again? And the obvious, the reason for that becomes relatively clear when you look at the right-hand side, where Yang plotted the precursor intensity of either those peptides that are identified in blue or in red, which they're not identified. And you can see that it somehow stabilizes here. You don't get any more identifications. And that is simply because the precursor MS-MS to be able to lead to a identification. So that says going beyond 3 hours really also doesn't make much sense, because the mass spectrometer doesn't have the sensitivity in order to detect the fragments from those precursors that the mass spectrometer will still subject to fragmentation, but then the ion current in the tandem mass spectra is too small. Now, what does that mean in terms of proteins and peptides? Again, looking across the different times on this axis here, on the left-hand side for peptides and on the right-hand side for proteins. And now I'm bringing in also our artificial intelligence tool called ProZ that is rescoring essentially max quant results to tease out more peptides and proteins. And what you can see is as the gradients get longer, you also get more peptides. And you're losing a certain amount of peptides simply because Max Quant and Prosit don't agree that they are actually confidently identified. But when you look around on the protein side, it's actually quite possible from a HeLa digest, I believe this is within 3 hours, to get about nearly 7,000 proteins out in a single shot. So that's encouraging. And then we basically took this to some application where we looked at Arabidopsis tissues, in this case roots, the human breast cancer cell line, mouse preparation of mouse small intestine or the thymus, and the story is kind of always the same. We actually get many thousand proteins, and the current record stands at nearly 9,000 for Arabidopsis roots, and that compares actually quite favorable to data from the Kuhn lab, where they coupled nano LC separations with FAMES gas phase separations, and in a single shot, also in 3 hours, managed to get nearly 7,000 proteins from whatever digest that they used here. So that is comparable to the figures that we're getting. So it seems that good chromatography can generate large numbers of quantifiable proteins. And again, I keep saying it, the micro LC system is not as sensitive as the nano, but it's actually not that bad. Okay, last slide on this part of the story is just looking again at the quantitative accuracy, and the long and short of it is that we do replicate analysis of these different tissues. Most of the CVs are below 20%, which again is good for quantitative precision. And then also if we ask the question, so what part of the proteome do we actually cover in a single shot? So what we've done is here just to plot out the abundance rank of these different samples, and then accumulate the amount of IBAC for getting an idea of how much of a particular protein contributes to the total. And then, of course, at the beginning, this sharply rises. And it's a typical observation that half of the total protein mass is contributed by only a handful of different proteins. But then you can see how this develops. And yes, there are some detailed differences in detail, but largely, essentially, this is the dynamic range we're getting. And you can plot this in a different way to get distributions of IBAC values. And you can see that the single shot method spans about 5 to 6 orders of magnitude dynamic range of detection. So that is again something that would also be very difficult to do in a single shot nano LCMS system, simply because you cannot load enough material. All right, so that was sort of the excitement around micro LC-MS, and my laboratory has converted at the moment 3 instruments in that mode of operation, and they are going through a very large number of samples, and we are not gonna, so we don't treat this as sort of a method development fun project, but we actually have made it an integral part of our analytical pipeline. So in the second pipeline we've just established, where we're following also the lead of other people, but with an interesting twist, is we have become more and more interested in analyzing clinical material that is formalin fixed, and that has traditionally been a bit hard to deal with, but a couple of improvements in the workflow that other people have made have really changed the situation. So for example, when you take macro dissected samples from a microscopic slide, of course, the first thing you have to do is de-cross-link this, and people have worked out protocols for doing that. Great contribution from Jeroen Kreisberg lab was the introduction of this SP3 workflow, which allows us to work with things like SDS. And then for these clinical applications, we've basically settled on using this EvoZep LC system simply because it has these disposable tips, so that we can deal with sample-to-sample carryover in a very pragmatic way. And what Yunjin and Stefan investigated, how this workflow could benefit from the addition of a FAMES gas phase separation in addition to the mass spectrometer. And that's what they spent some time on optimizing. And on optimizing the sample preparation, it became quite clear that that having higher tris concentrations in the de-cross-linking actually helps, that keeping the pH under control helps in order to revert much of the cross-linking. And then maybe more interesting, because it's kind of newer, is then we looked at the distribution of compensation voltages and how many peptides we would be able to identify given either a gradient of about 3/4 of an hour or 1.5 hours. And you can see here the characteristics of those CV values, and you can use that information to in silico ask the question, which compensation values should you combine in order to maximize the number of peptides you could detect. And here's just shown that the data matrix and the sort of the sweet spot in this particular case was 40 volts and 55 volts, which collectively should give about 30,000 peptides. But of course, this in silico analysis doesn't fully reflect the reality when you do the measurements. But nevertheless, when you analyze this systematically and ask the question, how many compensation voltages can we use within a single LCMS run and how many peptides does that give us? This obviously is a question of how much time you give the system. Switching between compensation voltages takes some time. So with short gradients, it doesn't really make any sense to use more than one compensation voltage. And if you use two, it doesn't harm too much either, but you don't gain anything. If you use slightly longer gradients, you only really see a benefit the moment you put on two compensation voltage settings in the same run, but then also the benefit is over when you run 1.5 hours of gradients. Sort of you can afford to run 4 or 5 CV settings in the same run. And what I want to point out is that FAMES does cost you some sensitivity, because these data points over here are the runs without any FAMES, and you can see the moment you switch on one compensation voltage in FAMES, the numbers inevitably drop. But then they recover if you do gas phase fractionation. And what is true for peptides is also true for proteins. So we basically have settled on a system that uses, I think, 5 compensation voltage settings within a 1.5 hour run. And if we do this, and that's sort of the middle column here, we can cover about 40,000 peptides from this particular sample using a total of 2 injections of 1.5 hours each, and injecting 600 nanograms in each of those injections. And we've compared that to a classical basic pH reverse phase fractionation of such samples, which actually don't do so well, even if you use 10 times or 20 times more material. So it seems the losses here outweigh the gains. But then if you bring in FAMES to that one, you actually outcompete FAMES only. But again, you need quite a bit more material. So essentially for our purposes is we've settled on this middle ground here where we get good coverage or reasonable coverage, but at very little total sample quantity requirements. And that helps us to deal with the clinical samples where sometimes there isn't a lot of it. And here's just to show on lymph nodes and prostates what the numbers are. And also there is some benefit from using max quant match between run feature. One always has to be a bit careful how one uses this in order to not generate too many false positives, but that's been covered in the scientific literature elsewhere. So, and I think I only have 2 minutes left. If I look at my watch here, just two minutes on another. All right. All right. So, but still, so third and last topic, which I'm only going to touch on briefly, is something we call the Proteome Tools project, where we have, together with the peptide company JPT and also with Thermo, analyzed more than 1 million synthetic peptides and generated hundreds of millions of MS spectra in order to generate essentially spectral libraries for many uses. And these peptides cover unmodified peptides, and the idea really was to generate this collection of spectra one could use, we or others could use to learn something from. And it's very gratifying to see that somebody posted this a while ago, the list of 100 most downloaded projects from Pride, and the top number 1 is Proteome Tools. So it seems people are downloading this, and that's of course great to see. And then we have a second tranche of data released, and in general, all of those arrows essentially represents work from my lab, and we do believe in data sharing for the benefit of others, and it's nice to see that people apparently use our data, so that's good. Sort of pointing you to to the talk of Matthias Wilhelm later in the week, is what have we done with this? And one of the ideas was, can we learn from such data sets what the tandem mass spectra of peptides should actually look like? And here's an extreme case where this is this particular singly charged peptide that isn't tryptic, and it produced, from a synthetic peptide, it produced this spectrum. Sorry, that was the reference spectrum in the, yeah, so that's the standard peptide that we made. And in some experiment, we initially found it. And the mass species among you might say, this is a terrible looking spectrum, but actually when you make the synthetic peptides, that's what it looks like. And Matthias and people around him have built a deep neural network to be able to learn what those fragmentation spectra should look like. And we called this PROSIT and published this about a bit more than a year ago. And that turned out to be a really fun project, because it looks like now we're looking at a tryptic peptide, we can actually predict these spectra with a ridiculous amount of accuracy. And this is what is shown on the right-hand side here, is when you ask the question, how well do the spectra of the experiment and the predictions agree, then PROSIT sort of gets it right with a median Pearson correlation of better than 0.99. And when we usually use the spectral angle as a measure because it's a little more sensitive. But essentially what it says, these tryptic peptides can be predicted with an amazing level of accuracy. And the same is true actually also for retention times. And here's a well-known retention time model called SSR-Calc from Oleg Krokin's lab that really already a while ago showed that retention times of peptides can be predicted pretty well, but PROSIT does an even better job here. And so we've stopped putting more time into this because we think it's now kind of good enough. And that enables a couple of things. And maybe one of the most exciting things is that we can basically get a 10 to 100 times lower false discovery rate on actually identical data, simply because PROSIT can rescore these tandem mass spectra with the question of, does the spectra actually support the identification, and essentially using retention time where it should be, is it where it should be, and looking at the intensities of the fragment ions and asking the questions, is the distribution of intensities of these fragment ions as we would expect. And that is a very sharp knife together with a tool called Calculator that was generated by Lukas Schell's laboratory in Sweden to come up with these criteria that score identifications. And essentially the long story short is we can tighten up the false discovery rate requirements without losing much data at all. And that has advantages for a number of applications. For example, although I'm not showing it here, in the field of microbiome research, where you have thousands of different species together in one sample, so you have to consider a lot of potential genomes to search against, and this is where the classical false discovery rate estimations really kill you, and tools such as PROSIT can really help there. But more on that in Matthias's talk on Friday, but here ending just with another sort of gratifying observation is that when we put PROSIT onto our public website, Proteomics-DB, we actually saw that people began using it, and the fun fact is here, when you track that over time, the fun fact is here is somebody posted about PROSIT on a blog spot post on Proteomics News, and look what happened. It seemed nobody had really seen this before, but when that posting happened, the number of predictions that were requested at our website began to rise sharply, and we also integrated PROSIT within Skyline, and we can also see that the Skyline community is using this, and this I haven't updated in the last 2 months, but PROSIT has made billions of spectrum predicted via Proteomics-DB for the community, and Matthias has sort of converted this into, if you were to do this empirically, you would have to expend about 100,000 hours of LCMI's time to get those spectra. So that's nice to see that this tool is being used. And with that, sort of I'm coming to the end. I spent quite a lot of time on this micro LCMS system because I really would like you to consider doing this. It makes proteomics also more accessible to people. Nano has its problems and needs special equipment, whereas micro LCMS doesn't need so special equipment. I spent a little less time on the FFPE, but I think this is actually also a combination that works well. And others could, of course, too. And I didn't really spend much time on ProSit, but as I said now, 3 or 4 times, you're going to hear much more of it later. And with this, I'm at the end, and I should extend special thanks to Yang Bian, who really made this micro LCMS possible, Stefan, Eckert, and Nün for the famous work on FFPE, and Matthias and Tobias Schmidt and teams have made ProSit possible. And again, I have a wonderful team of people with whom I work every day, and they do all the work, and I get to talk about it. So with that, I'm closing, and I hope I had something interesting to contribute to your workshop, and I'm happy to take questions.
Mr. Kuster, thank you so much. It was really wonderful, and I'm sure very eye-opening numbers, and the kind of data which you showed from the micro LCs, I'm sure going to be game changer. Lot of people are going to adopt that and going to utilize the power of these chromatography to really build the deep proteome coverage. And I just want to tell all our workshop participants that many of them are independent investigators setting up their own labs for proteomics. I think there is considerable amount of time, effort, and patience goes into method development part, irrespective of whether you like it or not. Even if you are deep in biology, you have to spend still considerable amount of time in method development. What is best suited for your sample type and your projects, especially looking at the gradients, the run hours, and kind of the column usage. All of these things are unavoidable part of this game, and you have to be doing this thing towards your project. And that's why I think a lot of time and intense effort goes into doing the proteomics. We are very also privileged to have on Friday Matthias Wilhelm, who will talk about ProSit, and the workshop participants can definitely make use of the hands-on session which is going to conduct on this software. And you have seen the very beautiful numbers, how the community has adopted that and using that. With this, let me thank Professor Kuster for this wonderful lecture, and open for the forum for the questions and discussions. Open to all of you now. All right, first hand comes from Dr. Sudarshan. Please go ahead.
Good afternoon, sir. It was really very interesting to hear you, and lot of data-based information. At many occasions, honestly, I was not able to understand also many of the things. But it was very good. I appreciate it. But 3-4 years ago also, we had heard some lectures from systems biology. They were also working on the importance of micro LC based in comparison to nano LC. So I wonder, and I would like to hear from you, that why the global scientific community now are more inclined to use nano LC based nano LCMS, in spite of all these good results from micro LC.
Yeah, so I think the breakthrough in proteomics actually came with the ability of dealing with what at the time was considered very small quantities of samples, and therefore you had to downscale everything in the sample preparation, but as well as the chromatography. And basically that breakthrough made a lot of people follow that lead, and then that stuck with people. But then there's, of course, and then it's hard to change. That is also true. When you set up your lab to do nano LC, then and you've mastered to do this, then you stick with it, because going away from it at minimum needs another investment into equipment, and it needs an investment in time for people to try. But I think it's swinging back now, because particularly for applications where sample quantities are not a problem. And we are not the first to do this, really. We can't claim that we are the first, but I saw a paper in analytical chemistry, I think 3 years ago now, by Juraj Lenčko from Prague. And I was fascinated because he showed that these peaks are so ultra sharp, and you can actually do some meaningful experiments with it. So we can't claim that we've invented this at all. It's just we jumped on it, and it worked so well that we never will go away from it. But again, the DIA people have sort of walked away from nano a little earlier, because they also realized that chromatography is important. And some people I know who work with body fluids even use the classical 2.1 analytical columns. And of course, they will need even more sample, but they're going to have even better chromatography. But for body fluids, it really doesn't matter. I mean, our protein plasma is at least 50 grams per liter, so that's 50 micrograms per microliter. So there is a lot of protein there. And also in urine, for example, 5 microgram of proteins from urine, I don't know, that's probably just 10 microliters of it also. So it's not a problem.
Yes.
So although and last word maybe on sensitivity, in the Nature Communications paper, we did show that it is also fine for affinity purifications and also phosphorylation. But I will say there is this factor 3 to 10 in sensitivity that the nano has an advantage. And if people want to go to very rare PTMs or rare cell types, even single cell proteomes, I mean, there's no way that big scale chromatography is going to be the suitable method.
Yeah, thank you.
Okay, wonderful. I think thanks for these comments. I'm sure participants are going to be very open who are all starting the labs, that what to go for, nano or micro LC. All right, Dr. Surekha Jingde, she is one of our proteomics scientist experts from India. Yes. She's next.
Professor Costa, I have 2 questions. How old blocks, FFP blocks, can one use for the study? And the second question is, how many sections do you really need to get reliable MS data?
Yeah. So the first question, I think, was about how old the blocks can be. Yes. We have not tested that, but I would imagine they could be ancient. The experience from a different area is from the gels, from protein gels. We analyzed proteins from gels even 10 years after the gels were run, and that was never a problem. But we have not tested this on the FFPE. But my gut feeling is it should actually be not a problem to run really old blocks as well. Then the second question.
Sorry, there have been concerns about how the material was fixed within the paraffin and what paraffin was used and so forth. So these are the concerns that come up.
Yeah.
And therefore the aging also would matter.
It could. I haven't got any data on this. Okay. As I said, it's just a gut feeling. The second is how much does one need? The data that I showed, I think, were performed on 4 slices of 10 micrometer each.
Okay.
But then it usually generates 10 or 20 microgram of protein, and then we only use 1 of them. So I think it's entirely possible for macro dissected samples to use a single slice. Let's say 5 by 5 millimeter is, I think, our standard. We haven't tried tissue microarray type of samples. That's on the list of things to try. That could become more challenging because they are much smaller. But yeah, just for reference.
Depends on the size of the tissue in the block.
Also, of course, yes. And the heterogeneity. What we are also only beginning to learn, of course, is the tissue heterogeneity within a block will, of course, have an influence on what you get out from the proteomic experiment.
Correct. So everybody starts looking.
That is going to be still hard for some time.
Yes. Yes. Thank you so much. Yeah.
Great. Thank you. So now next question is from Argyo Banerjee. Argyo, go ahead.
Yes, so it was a nice lecture. I will come to the point. According to the slide, what you showed in Baker Jensen et al. Okay, you have taken the peroxide scores of 1% FTR for identification of the highly confident peptide. Now, each of these identification of the confident peptide depends upon like what factors, what variables you use. If I am going with the Orbitrap system, like Orbitrap kind of system, people say that you have to match it with the NIST library, like the MS Pep search and all that. You have to match with the available synthetic library for the collection of the Orbitrap. And if it matches, and if it has a score of less than 500, means more than 500, or an correlation score of more than 1, then you can tell that it is like good, like your peptides are highly identified, your peptide is. I have seen a score of here also, like the Andromeda score and the PROSIT score. So like as you have compared with the MaxQuant, like how your software is robust with the MaxQuant. Have you tried with the other commonly available search tools like means Proteome Discoverer, Spectranaut, and all that? And how is the sensitivity of your software as compared to all of those? Then it will be a great revolution of proteomics, because everybody is able to defend their data by their own side. But when we are running the raw list parallelly, we see that some peptides come in sequest-ST, some peptides come in Amanda, MS Amanda, some peptides come in, means they are not, like, it is not occurring throughout. This is the ambiguity.
Yeah. So clearly there is ambiguity, even if you filter by whatever criteria. 1% is still 1%, and and 1% of 100,000 peptides will mean that 1,000 peptides of them could be just there for random chance alone. And all of the search engines work using kind of different and similar ideas. So I don't know exactly what's under the hood of MS Amanda, and I don't know exactly what's under the hood of MaxQuant. But essentially what we do with ProzitXU, it's not a search engine. We're taking the results of a search engine and ask the question, does that identification that the search engine already made, is that plausible or not? And for that, we use the predicted and the experimental tandem mass spectrum and essentially compare them together along with all these scores provided by Percolator to come up with essentially a yes or no, whether or not they are within our 1% criterion. So to answer your question more directly, what frequently happens is that the rank 1 peptide of MaxQuant will not be the right one. We promote often the rank 2 because the spectral actually are a better match, and MaxQuant gives more weight to mass accuracy, but it doesn't use the intensity of the fragment ions. We have, together with Thermo, integrated Prozit into Proteome Discoverer, so that's something that all the Proteome Discoverer people can now use. And the results of Proteome Discoverer searches, or what under the hood of that is Sequest, is not identical to MaxQuant, and therefore also the rescoring results will not be identical. In that sense, the idea of uncertainty will always be there, but what I try to say is by extending the capabilities of search engines to the question of, or to the using the information of retention time and relative fragment ion intensities, this adds discrimination power. And it adds actually a lot of discrimination power, so that even if we didn't want any more peptide, we could tighten up the stringency of the FDR by a factor 10. Or conversely, if we still think that 1% of error is okay, then we're going to increase the number of things we have in our box.
All right, so this was Dr. Bernard Kuster. I hope you have all learned something new from this lecture. Essentially, while talking about proteomics, what data we generate for peptides are essentially being using, the nano-LC, nano-liquid chromatography, which we showed you in demonstrations in the beginning. But there is so much effort goes on to method development, where there is more and more emphasis about the reproducibility of data, as well as the coverage of the proteome, right? And that's where his first point was about using micro-LC, where you can load more amount of peptide and look at chromatographic peaks and look at how reproducible they are. And therefore, if you are looking for more label-free quantification type of analysis, you are probably looking for much more robust data if you were to use the micro-LC. And I'm sure his data convinced all of us that how well one could use, to increase the coverage drastically. So that is something which is worth trying out, and keep your mind open that the field of proteomics, looking at the complex proteome mixture and analytical methods, does require continuous investigations, and one need to be very open to adopt new methods. Second thing was regarding the FFP tissue blocks, which is the paraffin embedded formalin fixed tissue blocks. We also try to optimize in our lab, because given that in different cancer projects which we work on, with Tata Memorial, we get out of these tissue blocks where patient tumor biopsies have been stored in these paraffin blocks. And removing that paraffin material actually becomes very challenging. And we also many times face that the quality of material coming from different path labs or the hospitals may also be different. So therefore, again, a lot of optimization goes on to doing the FFP-based proteomics. Think about when some surgeons are, excising the tumor and putting that in liquid nitrogen right away. That is known as the flash frozen or the liquid nitrogen stored sample, which is the ideal sample for research. But that is not the possible sample all the time for clinicians. They are busy doing their surgery and the clinical part. So then most of the tissue actually being frozen and kept in the paraffin embedded material so that it can be used for the immunohistochemistry later on. Can we do proteomics on using these FFP blocks? And if we can, then probably we are talking about really huge number of samples available in a lot of tissue repositories. So we have optimized quite a bit of FFP-based proteomics as well in the lab. And for Shashwati, one of the TAs who is working with you on different analysis, she has done more work on the FFP. And as a result, now we are able to get a lot of sample from Tata Memorial for doing this research and use the FFP proteomics. Final point was regarding closet. When we are talking about looking at the data, I think many times what you are looking at essentially to avoid your complexity of your system configuration and your bandwidth and internet, we have not given you raw data to be analyzed. We have essentially given you process data, and but if you have an opportunity to have good bandwidth and a good system, or if you're in IT Bombay campus, I think you should also look at more raw data analysis. Then in that way, it will be important sometime if you're looking at, say, phosphorylation, where you want to make prediction for some peptides getting phosphorylated. So you need to cross-check the spectra, MSMS spectra, and find out that do you still see actually, on serine, threonine, tyrosine, these amino acids, are there actually changes happening? Do you see some Dalton change with the phosphate residue being added? So can you do that experimentally, validate these? And therefore, looking at the raw MSMS spectra becomes important. And PROSIT is definitely one of the very important tool of capturing these information. Whenever I will have time more, I will show you this session from Matthias Wilhelm, which will give you hands-on activity for the PROSIT. But given the class level and time, I had thought not to introduce you this software for the class activities. But it will be an important thing for you to learn and know that PROSIT is really important to validate your MSMS spectra. All right, that's pretty much it. I showed you this lecture and a couple of questions from the participants as well. Now, if you have any question from his lecture, I'll be happy to take right now for you to just give you some clarification. So please ask me if you have any specific question for Dr. Kuster's lecture, which was unclear to you. All right, if not, there will be some more questions in the video, which I will upload. You can watch those and understand it better. This was actually the cutting edge, more recent research-based stuff. If everything you're not able to follow up, it's still fine. I hope you got the flavor of the magnitude of effort goes on to look at the complex proteome analysis, and that's where this group was able to publish the draft human proteome map. They also make for all the human peptides, they made a synthetic peptide libraries and made it again publicly available. So all of these are very important contribution, which is actually helping the whole scientific community and proteomics community. You also seen the usage of proteome tool kit and so much importance, and now you see increased usage of the PROSIT. So all of these are publicly open. Please start using them if you intend to work in this area. Of course, even as a class student, you should be at least being aware that what are all resources available to all of you. So with this, let me end today's lecture. I'm feeling that the first group of the students, the last presenters, were really good. They really made good presentation. Of course, Nilesh was also good today, but the bar being raised so high that some of the students started feeling more nervous about it. Please don't do that, because every topic is different. Every analysis will be different. So you have to make sure that you have to meet your timelines well. Given the nature of the overall schedule what we have made, it will be difficult for us to accommodate requests last minute. I have given all the guidelines already to all of you. Please follow that, and we'll look for more exciting presentations from your projects in the next class. If you have any doubts and clarity which you need for your data analysis, please let us know. If required, we can actually have a short session with the next week participants on Monday evening, so that we can take your questions and we can make any clarifications. We are there to help you out, but you have to make sure that you please forget the last minute practice of doing the things and be more professional. Okay, I hope you're enjoying learning proteomics, and I'll be happy to take more and more questions on the chat or even directly by the email. That's all for today. Thank you very much, everyone.
Thank you, sir.
Welcome.
Thank you, sir.